Welcome to the deep dive. We take complex ideas and, well, we try to make them clear and compelling.
And today we're diving into something truly fundamental, proteins. Specifically, how their shape dictates everything they do.
Right. And sometimes the best way into these big biological topics is through real stories, real people.
Exactly. Let's think about some cases, maybe ones you'd encounter in, say, the waiting room.
Okay. Like Will S. He's young, but he keeps getting these episodes of terrible pain, joints, chest, he gets short of breath. What could be happening inside his blood?
Or Amy L.A. Doctors are puzzled. Her tongue is enlarged, she's got kidney and heart trouble, and biopsies are showing these weird protein clumps.
And then Diane A., she's managing her diabetes, and her doctor focuses on this one lab value, HbA1c. What's that really measuring?
It tells a story about proteins, too. And Ann J., she arrives in the ER, chest pain. They test for something called cardiac troponin T almost immediately. Why that specific protein?
Yeah, these are all situations where proteins are absolutely central. So our mission today is to unpack this connection. How do proteins build from just, 20 standard amino acids?
Just 20 basic building blocks, yeah.
How do they fold into these incredibly precise 3D shapes that let them do specific jobs? And crucially, what goes wrong when they don't fold right, leading to conditions like the ones we just mentioned?
We're going to journey from that basic amino acid sequence all the way up to these large multi-part protein machines. We'll look at folding, how they grab onto other molecules.
And connect it all back to diseases like sickle cell, amyloidosis, and others. Get ready for some aha moments, hopefully.
Definitely.
Okay, so proteins. Workhorses of the cell. You hear that all the time. They do everything. But how? How do they get this amazing versatility from only 20 amino acid types? Where's the starting point?
It really does all begin with the sequence. That's level one. The primary structure. It's just the linear order of amino acids linked end-to-end by peptide bonds. Think of it like beads on a string in a very specific order.
And that order isn't random, right? It comes from our genes.
Exactly. The genetic code dictates that sequence precisely. And that sequence, that primary structure, is the absolute blueprint. It determines all the folding, the final shape, the function. Everything follows from that initial string.
Okay, so you have this string, this polypeptide chain. Does it just flap around like a wet noodle?
No, definitely not. Almost immediately, it starts to organize itself into local repeating patterns. These are the secondary structure.
Local patterns? What holds those together?
Hydrogen bonds. Tiny, relatively weak bonds, but lots of them, forming within the polypeptide backbone itself. They act like little internal magnets, pulling nearby parts of the chain into specific shapes.
And what kind of shapes do we see most often?
Well, the two big ones are alpha helices and beta sheets. An alpha helix is, imagine a spring or maybe a spiral staircase. It's a really stable, rigid coil.
Okay, the coil.
Yeah, and it's efficient for packing the chain. The hydrogen bonds form in a very regular pattern along the spiral, and the side chains, the unique parts of each amino acid, stick outwards.
So they don't bump into each other.
Right, avoid steric hindrances. But here's a cool detail. One amino acid, proline, it's kind of famous, it's called a helix breaker.
A helix breaker? Why?
Its structure is unique. It has this ring shape that just doesn't fit properly into the smooth spiral of the helix. It kinks the chain and disrupts the hydrogen bonding pattern. So if you see a proline, chances are the helix ends or bends there.
Wow, one amino acid makes that much difference. Okay, so helices are coils. What about beta sheets?
Beta sheets are different. Think less coil, more. Like a folded sheet of paper, pleated back and forth. A zigzag structure.
Okay.
These form when different segments of the polypeptide chain lie side by side, and hydrogen bonds form between these adjacent strands. The strands can run in the same direction that's parallel or opposite directions, which is antiparallel. That's actually more common.
And the side chains?
They stick out alternately, sort of above and below the plane of the sheet.
Got it. So you've got these local folds, helices, and sheets, maybe some loops and turns connecting them.
Exactly. Those less regular bits are important too, often on the surface, providing flexibility.
And all these secondary bits fold together to make the protein's overall final shape.
Precisely. That's the tertiary structure. It's the complete three-dimensional folding of the entire polypeptide chain, incorporating all those alpha helices and beta sheets. This is the protein's unique functional shape, its native conformation.
The native conformation. And what does that typically look like for, say, proteins floating around in the cell?
Well, many of the proteins we'll talk about are globular proteins. They tend to be roughly spherical, compact, and usually soluble in water.
And how are they arranged internally? Is it random?
Oh, not at all. There's a very clever logic. They typically have a dense, hydrophobic core. All the amino acids that dislike water, the nonpolar ones, get buried on the inside, away from the watery environment.
Hiding from the water.
Exactly. And the amino acids that like water, the polar ones, are usually found on the surface, interacting with the cell's aqueous surroundings.
What holds this whole 3D blob together? More hydrogen bonds.
Those play a role, definitely. But it's a combination of forces. The hydrophobic effect, that burying of nonpolar groups, is a major driver. Then you have ionic interactions, like salt bridges between charged groups, van der Waals forces, which are weak attractions between atoms close together, and yes, hydrogen bonds between side chains or with water. Sometimes you even get stronger covalent disulfide bonds between cysteine amino acids, locking parts of the structure together.
So this final 3D shape isn't just aesthetic. It has to be right for the protein to do its job.
Absolutely critical.
Yeah.
The tertiary structure creates the specific binding sites for other molecules. It has to have the right amount of flexibility in some areas and rigidity in others. Its surface needs to be suitable for its environment, soluble in water, or maybe embedded in a membrane. It needs to be stable enough to last, but also degradable when it's time to be removed.
And sometimes big proteins are kind of modular, like different sections do different things.
Yes, that's a great point. Large proteins often consist of distinct structural domains. These are regions that can fold up independently, almost like separate little globules connected together. Often, each domain has a specific function, like binding a certain molecule or catalyzing a reaction.
Okay, so primary is the sequence, secondary is local folds like helices and sheets, tertiary is the overall 3D shape of one chain. Is there another level?
There is. The highest level, quaternary structure. This is when you have multiple separate polypeptide chains, or subunits, associating together to form one large functional protein complex.
So teamwork.
Exactly. Think of hemoglobin again. It's not one chain. It's 4 subunits working together, 2 alpha chains and 2 beta chains. That's its quaternary structure.
Can the subunits be identical or are they always different?
Both happen. If the subunits are identical, we call it a homer, like in some enzymes. If they're different, like hemoglobin's alpha and beta chains, it's a heteromer.
Why bother with multiple subunits? Why not just make one giant polypeptide chain? What's the advantage?
Several key advantages. It often increases stability. It allows for complex regulation, like the cooperative binding we'll see in hemoglobin that's huge. And you can create much more intricate binding sites by bringing different subunits together. Think of antibodies, immunoglobulins. Their binding sites are formed where different chains meet.
Okay, that makes sense. So these intricate shapes aren't static sculptures. They do things, they interact. Let's get into the action, how they bind to other molecules.
Right, because that binding is their function, essentially. The molecules they bind are called ligands. A ligand could be a small molecule like oxygen or ATP or even another protein. And the binding is usually highly specific.
Like a key fitting into a lock.
Very much like that. The shape and chemical properties of the protein's binding site are precisely complementary to its specific ligand.
Can we measure how tightly that key fits, how strong the binding is?
Oh yes, absolutely.
Okay.
We quantify it using constants. The association constant, Ka, tells you how readily the protein and ligand bind. The dissociation constant, Kd, tells you how readily they fall apart.
So they're inversely related.
Exactly. A high Chi means strong binding. A low Kd also means strong binding because they don't dissociate easily. So if you have 2 ligands, A and B has a Kd that's, say, 100 times lower than A's Kd, that means B binds 100 times more tightly to the protein. Tiny numerical differences can mean huge functional differences in the body.
Let's make this concrete. Hemoglobin and myoglobin both bind oxygen, right? But you said they have different roles.
Perfect example to illustrate structure function, especially the impact of quaternary structure. Myoglobin is simpler. It's a single polypeptide chain, globular, found mainly in muscle tissue. It has one binding site for oxygen. Its job is basically oxygen storage.
Holding on to it until the muscle really needs it.
Precisely, like a little oxygen reserve tank. Hemoglobin, on the other hand, is the oxygen transporter in our red blood cells. And structurally, it's a tetramer, 4 subunits, remember? 2 alpha chains, 2 beta chains. Each subunit looks quite a bit like myoglobin, actually, and each carries 1 oxygen binding site. So 4 sites in total per hemoglobin molecule.
4 sites versus 1. Okay. And what's actually doing the oxygen binding in both?
It's a specialized helper molecule called heme. It's a non-protein component, what we call a prosthetic group, tucked into a pocket in both myoglobin and each hemoglobin subunit. Heme contains an iron atom, specifically iron in the Fe2+ state, right at its center. That's what the oxygen molecule physically binds to.
So the protein part holds the heme in place, and the heme iron grabs the oxygen.
You got it. A protein with its necessary prosthetic group, like heme, is called a holoprotein. The protein part alone, without the heme, is the apoprotein.
Okay, so oxygen binds the iron. Does anything happen to the protein structure when that occurs?
Yes, and this is crucial. When O2 binds, it actually pulls that Fe2 plus iron atom slightly, shifting its position within the flat heme ring. This tiny movement of the iron then tugs on an adjacent amino acid, a histidine residue, which is connected to one of the protein's alpha helices.
So like pulling a string that moves a lever?
Kind of, yeah. In myoglobin, this little tug doesn't do much functionally. But in hemoglobin, because of its 4 subunits interacting, this small shift triggered by oxygen binding to one subunit has major consequences for the other subunits.
Ah, this sounds like it explains that difference in how they bind oxygen. Myoglobin's binding curve is simple, but hemoglobin's is that weird S shape. Why?
That S shape is the hallmark of positive cooperativity. It's a fantastic feature enabled by hemoglobin's quaternary structure. When the first oxygen binds to one heme in hemoglobin, the resulting conformational shift makes it easier for the second oxygen to bind to another subunit.
Easier.
Yes, the protein shifts from a low oxygen affinity state, called the tense or T state, towards a high oxygen affinity state, the relaxed or R state. Binding the second oxygen makes it even easier for the third, and the third makes it easier for the fourth.
So the more oxygen it binds, the stickier it gets for the next oxygen.
Exactly. It involves breaking some interactions, some salt bridges between the subunits, which requires a bit of energy initially, but overall it allows hemoglobin to be incredibly efficient. It can readily load up with oxygen in the lungs where oxygen is plentiful, and then easily release that oxygen in the tissues, where oxygen levels are lower and the affinity drops back down. That S-curve reflects this transition from low to high affinity as oxygen binds.
That's incredibly clever. So it fine-tunes its oxygen grip depending on the environment. Are there other things that influence how tightly hemoglobin holds oxygen?
Yes, several important regulators help ensure oxygen gets delivered where it's most needed. One is a molecule called 2-ip-3-bisphosphoglycerate, or 2-ip-3-BPG. It's made inside red blood cells.
2-pp3-BPG, what does it do?
It binds right into a central cavity in the hemoglobin tetramer, but only in the T state, the low affinity state. By binding there, it stabilizes the T state, making it harder for hemoglobin to shift to the R state. The effect. It lowers hemoglobin's affinity for oxygen.
Why would you want to lower the affinity?
Because that promotes oxygen release in the tissues. It helps hemoglobin let go of oxygen where cells are actively metabolizing.
Okay, makes sense. What else?
Protons. Hydrogen ions, H plus X. This is known as the Bohr effect. When tissues are working hard, they produce metabolic acids like lactic acid and also carbon dioxide, which forms carbonic acid in the blood. This lowers the pH, meaning more protons around.
Right, more acidic.
And hemoglobin can bind these protons. When it does, particularly at certain histidine residues, it again stabilizes the T state and lowers its oxygen affinity. So in active acidic tissues, hemoglobin is prompted to dump even more oxygen. Then, in the lungs, where CO2 is breathed out and the pH is higher, hemoglobin releases the protons its affinity goes up, and it readily binds oxygen again.
It's like it senses where the metabolic activity is highest.
Precisely. And carbon dioxide itself can also directly affect hemoglobin. CO2 can bind to the N-terminal amino groups of the hemoglobin chains, forming carbamate groups. This also stabilizes the T state, further encouraging oxygen release in the tissues where CO2 levels are high. It all works together beautifully. It really is an amazing system. Okay, so we've seen the beauty of protein structure when it works, but what happens when it goes wrong? When the folding messes up, that's where we see the connection to disease. Even small errors in folding or structure can have devastating consequences. Remember, it's the primary sequence that should dictate the final, stable, native conformation.
But proteins can lose that shape, right? Denaturation.
Yes, denaturation is the loss of secondary, tertiary, and maybe quaternary structure. Things like heat, extreme pH changes, or certain chemicals can disrupt those non-covalent bonds holding the protein together, causing it to unfold and lose its function. Think about cooking an egg. The heat denatures the albumin protein, turning it white and solid.
Can they refold, go back to normal?
Sometimes. Simple proteins, under the right conditions, can spontaneously refold or renature. But for many complex proteins, especially if their primary structure has been modified after synthesis, like insulin, which has a piece cut out, renaturation might be impossible. And often, folding isn't spontaneous in the crowded environment of the cell. Proteins often need help.
Help? From what?
From other proteins called chaperonins. Think of them as protein folding assistants. They bind to newly made polypeptide chains or proteins that have started to misfold, and they use energy, usually from ATP, to help guide them into their correct native conformation, preventing them from aggregating or getting stuck in wrong shapes.
Like quality control for protein folding.
Exactly, essential quality control.
Before we get deep into the misfolding diseases, maybe a quick word on a different type of protein, not globular, but fibrous, like collagen.
Good idea. Collagen is a great contrast. It's the main structural protein in our connective tissues. Skin, bone, tendons. It's incredibly strong. Instead of folding into a globule, it forms a long fibrous structure specifically, a triple helix.
Triple helix, 3 chains wound together.
Yes, 3 polypeptide chains called pro-alpha chains wrap around each other. Its amino acid sequence is also unusual. Very repetitive, rich in glycine and proline. But what's really key for collagen stability is a modification after the chains are made.
What kind of modification?
Certain proline and lysine residues need to have hydroxyl groups added to them. This process, called hydroxylation, absolutely requires vitamin C as a cofactor for the enzymes involved.
Ah, vitamin C.
Yes. Those added hydroxyl groups are crucial for forming hydrogen bonds that stabilize the triple helix. Without enough vitamin C, you can't properly hydroxylate collagen. The triple helix is unstable, and connective tissues weaken. That's the basis of scurvy.
Wow. Directly linking diet, protein modification, structure, and disease. Hey, let's bring it back to our patients. Will S. and sickle cell anemia. How does his protein structure go wrong?
Will's case is a textbook example of a single amino acid change causing disaster. It's all about his hemoglobin, specifically the beta-globin chains. He has a mutation where just one amino acid is wrong. A glutamic acid, which is hydrophilic, water-loving, is replaced by a valine, which is hydrophobic, water-hating.
One single change out of hundreds of amino acids.
Just one. But this valine substitution creates a hydrophobic sticky patch on the surface of the hemoglobin molecule, but only when it's deoxygenated. We call this abnormal hemoglobin HbS.
A sticky patch. What does it stick to?
It sticks to a complementary hydrophobic pocket on another deoxygenated HbS molecule. So when oxygen levels are low, like in the peripheral tissues or during exertion, these HbS molecules start sticking together, polymerizing into long, rigid fibers inside the red blood cell.
Polymers inside the cell. That sounds bad.
It is. These stiff fibers distort the red blood cell, forcing it out of its normal flexible disc shape into a rigid crescent or sickle shape.
And what does that do to Will?
Those sickled cells are terrible for blood flow. They're not flexible, so they get stuck in small capillaries, blocking blood flow and oxygen delivery to tissues downstream. This causes ischemia, leading to the intense pain crises Will experiences, damage to organs over time, and chronic anemia because these fragile cells are destroyed more rapidly. It's fundamentally a disease of abnormal quaternary structure aggregation and reduced solubility.
Such a cascade from one tiny change. Okay, let's shift to Amy L. and her amyloidosis. You mentioned protein clumps?
Right. Amyloidosis isn't just one disease. It's a group characterized by the buildup of abnormal, insoluble protein fibrils called amyloid in the spaces outside cells.
So not inside the cell like sickle cell, but outside.
Correct. Extracellular deposition. And these amyloid fibrils have a very specific characteristic structure, regardless of which protein is misfolding to form them. They are rich in beta sheets, arranged perpendicular to the axis of the fibril. Think of it like stacking beta sheets on top of each other.
And that structure is detectable.
Yes, diagnostically. When tissues with amyloid are stained with Congo red dye and viewed under polarized light, they exhibit this classic apple green birefringence. It's a key sign.
In Amy L.'s case, AL amyloidosis, what protein is misfolding?
It's derived from immunoglobulin light chains, pieces of antibodies that are normally produced by plasma cells. In her case, these light chains are misfolding, aggregating into those beta sheet-rich amyloid fibrils, and depositing in organs like her tongue, kidneys, and heart, causing them to enlarge and malfunction.
And you mentioned a seeding idea earlier.
Yes, that's a really important concept in amyloid and similar diseases. Once a few misfolded proteins aggregate, they can act as a template or a seed, accelerating the misfolding of other normal copies of the same protein. It creates a chain reaction of misfolding and aggregation.
That sounds almost infectious. Can a protein itself spread disease like that?
In a way, yes. That brings us to prion diseases. Prions are fascinating and, frankly, quite scary. They stand for proteinaceous infectious agents.
Protein infectious agents.
Exactly. These diseases involve a normal cellular protein called PRPC, prion protein cellular, which is usually rich in alpha helices. But this protein can misfold into an abnormal disease-causing shape called PRPSC, prion protein scrapie, which is much richer in beta sheets.
So similar to amyloid, a shift to beta sheets. But how is it infectious?
Here's the disturbing part. The misfolded PRPSC protein can interact with normal PRPC proteins and somehow induce them to misfold into the PRPSC shape too. It acts as a template, converting normal protein into the abnormal aggregation-prone form.
It converts them like a zombie protein.
That's actually not a bad analogy. This conversion sets off a cascade. The PRPSC aggregates into clumps that are resistant to breakdown by cellular proteases, accumulating particularly in the brain and causing neurodegeneration. Think of Creutzfeldt-Jakob disease, CJD, or mad cow disease. It's a disease propagated solely by a misfolded protein template.
It's truly unnerving. Okay, let's move to something maybe less dramatic but still impactful. Diane A, her diabetes, and that HbA1c number.
Right. This involves a different kind of protein modification, not misfolding per se, but non-enzymatic glycosylation.
Glycosylation, adding sugar.
Exactly. But non-enzymatic means it happens spontaneously without an enzyme directing it. Glucose circulating in the blood can chemically react with exposed amino groups on proteins, like the hemoglobin inside red blood cells or collagen in blood vessel walls.
Just sticks on.
Pretty much. And the rate at which this happens is directly proportional to the concentration of glucose in the blood. If blood sugar is high, more glucose sticks to proteins.
And this is irreversible.
Yes, the initial attachment forms a shift base, which then rearranges into a more stable product. Because red blood cells live for about 120 days, measuring the percentage of hemoglobin that has glucose stuck to it, that's HbA1c, gives doctors a picture of the average blood glucose level over the past 3-4 months.
So it's not just a snapshot like a fingerstick glucose test, but a long-term average.
Precisely. It's incredibly valuable for monitoring how well diabetes is being managed over time.
Does the sugar sticking to proteins cause problems beyond just being a measurement?
Oh, definitely. Over time, these initially glycosylated proteins can undergo further reactions and cross-linking, forming complex structures called advanced glycosylation end products, or AGs. These AGs accumulate, especially with aging and poorly controlled diabetes, and they contribute significantly to the long-term complications damaging blood vessels, kidneys, nerves, the heart, partly by altering the structure and function of proteins like collagen.
Another way protein structure gets compromised. Okay, one last case. Ann J's heart attack and the cardiac troponin T-test. Why is that protein so specific?
This highlights the use of protein isoforms in diagnoses. Many proteins exist in slightly different versions, or isoforms, in different tissues. They perform the same basic function, but have slightly different amino acid sequences, usually due to being coded by different genes or alternative splicing of the same gene.
So there's a heart version and a muscle version of troponin T.
Exactly. Troponin T is part of the muscle contraction machinery. But cardiac troponin T, CTNT, found in heart muscle, has a unique sequence, different from the troponin T found in skeletal muscle.
And that difference allows for a specific test.
Precisely. Diagnostic tests, typically immunoassays using antibodies, can be designed to be highly specific for the cardiac isoform. So, when heart muscle cells are damaged, like during a heart attack, they release their contents, including CTNT, into the bloodstream. Detecting elevated levels of CTNT is therefore a very sensitive and crucially specific indicator of heart muscle injury.
Much better than older markers that might come from skeletal muscle too.
Far better. It allows for rapid and accurate diagnosis, which is critical for guiding treatment for Ann J and others like her. It's all down to subtle differences in protein primary structure between tissues.
Wow. What an incredible journey, really, from a simple string of amino acids to these complex machines and devastating diseases when things go awry. That primary sequence really is the code for everything.
It truly is. Every helix, every sheet, the final fold, how it binds oxygen, how it provides strength like collagen, it all traces back to that initial sequence. And as we've seen with Will, Amy, Diane, Anne, when that structure is compromised, whether by a mutation, misfolding, or even just chemical modification like glycosylation, the impact on health can be immense.
It really drives home how a protein's shape is its function, its destiny.
Absolutely. The native conformation is key.
So maybe a final thought for everyone listening. Next time you hear about a disease, especially one that seems complex or maybe has a genetic component, ask yourself, could this be, at its heart, a story about a protein? A protein that's folded incorrectly or can't bind its partner or maybe sticks together when it shouldn't?
It puts a different lens on things, doesn't it? Understanding this molecular level really opens up our view of health and disease. It's the intricate dance of molecules that underlies it all.
It certainly does. We hope this deep dive has illuminated some of that incredible world of protein structure and function for you.
It's been great exploring it with you. Thanks for joining us.